So we're still on problem solving questions. This is from an experimental design question from the 2018 paper, question 10. Okay, our context. An experiment carried out to investigate the evolutionary relatedness of 4 species of fish by comparing proteins extracted from them. Okay, so you're told that the more closely related species are, the more proteins they have in common. You're told that the sample of tissue was taken from each species of fish, heated in a solution to extract proteins. Protein extracts were then analysed by gel electrophoresis, and you're told that it extracts protein or separates proteins according to their mass. Protein standard containing proteins of known masses was also analysed. You're going to need that to be able to kind of read across, and the results are shown in the diagram. Each band is a protein. So here's our gel picture. We have the direction of movement of proteins going down this way. We have our 4 species, and we have the bands that have been created as they've kind of run down. What you've also got is the standard. So here's your standard here, and they're telling you the mass of the protein in protein standard in kilodaltons. You don't need to worry about that, but that's what that unit is there. And then we've got our distance travelled. So the only thing I would say to point out in this one, look, the 16, the smallest thing, is actually down here at the bottom. So there's your 16 band, 50 millimeters down. There's your 20, there's your 26, your 44, your 66, and your 108. Okay, because that only travelled 10 millimeters. Okay. Okay, identify 2 variables related to the protein extraction not already mentioned, which should be kept constant so a valid conclusion can be drawn. Okay, so what do we actually have about the extraction? Here we go. A sample of muscle tissue was heated in a solution to extract proteins. That is not a lot. So what you're going to have to give, I would go with sample of muscle tissue. You need to have the same mass. It needs to be from the same place. All that would be reasonable to look at for variables. Heated, what temperature? A solution. It's fine enough to keep it as a solution, but concentration of solution, volume of the solution, that could also be variables you would need to keep constant. Okay, during the preparation the samples were heated. This unfolds the proteins, changing their 3-dimensional shape. Name 1 type of bond that could have been broken to cause this change. Right, now there will be, if you're doing chemistry you might know more of these, but the one that you're expected to know would be hydrogen bonds. So hydrogen bonds, you also have ionics, you have hydrophobic interactions, so you could go for hydrophobic. Specifically you will have disulfide bridges, which are covalent links. Oh, bridges. You also have all of the van der Waal interactions. Now that actually includes hydrogen bonds, and if you're doing, as I say, chemistry, London dispersion forces is also a reasonable one to say. I would say for secondary structure, hydrogen bonds is the one I'm expecting to see. Okay, what you must not say is peptide, because if you say peptide bonds, that's not to do with their 3-dimensional shape, that's to do with the initial single line that you get of amino acids joined together. So if you break the peptide bonds, you're actually shattering the entire protein, not just changing its shape. Okay. Okay, we've got a graph to draw the distance travelled by the protein in the gel against the mass of the protein in the protein standards. Okay, additional graph paper if required. Fair enough. So what we need to do is what we changed versus what we measured. What we measured was our distance travelled, so distance travelled goes on this scale. You just put the whole thing down there. I'm just putting wee squiggles because I've not given myself enough space. This one, you're going to write the whole thing down here along the bottom, this bit in here, and then you just need to pick scales. Both of these are reasonable to start at 0, or you could start them at 10, but I would say just if there's enough space. So that's if this is the mass, we need to get up to 108, so 10, 20, this will be okay. 90, 100 and 110. Okay, going up the side, if we take that at 0 and we go up in 5s, Okay, that's 1 point. That's your axes with, as long as you've written absolutely everything in the label, and your scale. Okay, your second point is for the plot. All of these I think are going to be okay to plot. There's nothing that's going to be on a half box. So it's expected that everything is going to be on a cross point of a line, and then you join the dots. I'm not going to draw this one out because it's just much more footery on this, better on paper. Band X travelled 28 millimeters. Use the graph to identify the mass of the protein in band X. Okay, so this will depend on your graph. Depending on how easy they think it is to get a nice neat line between, you might have very little give in the Mark scheme. I can't remember, so we'll check it when we get to the end. But it's from your graph, so go along to 28 millimeters, run it up, and then run it across to get the mass. Okay, a quick calculation. We've got each species of fish contains a protein with a mass of 66 kilodaltons. Each amino acid has an average mass of 0.12. Calculate how many amino acids would be expected to be in this protein. Not exactly a complex calculation. It's just that it's not stuff you would normally see. So our total is 66, and our every amino acid is 0.12. So 66 divided by 0.12, oh, sorry, I typed in an extra 0, gives me 550. That's it. Okay, explain why it was concluded that species 1, 2 and 4 are more closely related to each other than they are to species 3. So here was the context, if you remember, and it said in here, the more closely related species are, the more proteins they have in common. Here's our results. So here's species 1. Species 1 has common with all of these at this point. It has these in common with 2 and 4, and these in common with 2 and 4, and this one down here in common with 3. So why are 1, 2 and 4 more closely related to each other than 3? Look at 3, it doesn't have many bands in common. Okay, and you have to explain that. Here's your Mark scheme. So this was the point I was saying about the graph. It says 32 kilodaltons is what they're expecting, or whatever the plotted graph shows. So if you've written something that isn't 32, then you go back to the graph, they will look at your graph and they will look to see if it actually agrees with that. Okay, you will notice there is an awful lot of knots here. So be very careful that you have actually read that one carefully. Okay, and that's that one.